Hi, everybody. This is Jason Key at SBGrid in Boston, Massachusetts. It is my pleasure to welcome Woody Sherman. So Woody is the Chief Scientific Officer at Silicon Therapeutics, based here in Boston, Massachusetts. So Woody came up at MIT, did his PhD there, and from there he's been in industry primarily, both at, I think it was at Biogen for a short time, then Schrodinger, and then I don't know if we can call Silicon Therapeutics a startup anymore, but started up Silicon Therapeutics, which is growing like crazy. And today he's going to tell us about work to come out of Silicon Therapeutics, a physics-driven discovery of SNX281, a novel small molecule STING agonist in clinical trials for solid tumors or lymphoma. So go ahead and share your screen.
Great, thank you, Jason. Okay, great. Well, thanks everybody. Thanks for joining. And I think there's a Q&A section to type in things that we could address at the end. But if there is any thing that's needed during the course of the presentation, feel free to just speak up and ask for anything where clarity is needed. So a little bit of an overview of Silicon Therapeutics for people who are not familiar with us. We're a fully integrated drug discovery company. We have everything we say from chip to clinic. We have supercomputers to run our simulations, a lot of GPUs and CPUs that we have in-house and cloud, and we actually do custom hardware research. We have a very strong simulation team where we've built out our own force field, small molecule force field, lots of simulation infrastructure that I'll talk about. And then the scientific expertise, which is a combination of roughly 50% experimentalists and 50% computational folk that we've all brought together under one umbrella. And I'll be talking today about how that's all worked and how that resulted in our first clinical asset and what we're working on next. So we're right down in the Seaport area, 451 D Street, right across from the convention center. If people have been down in that area, it's an old warehouse that's shown up there on the left that's starting to get converted into lab space. And so we have about 10,000 square feet down there. We have biology, chemistry, biophysics, structural biology, and plus all of our office space all right there. So really great location, very easy to get in and out, and plenty of room for expansion down there. People may have seen the news. We are now part of Royvance Sciences. It's really an amazing organization, highly distributed organization. So we are going to maintain who we are as Silicon Therapeutics, but be involved in this broader Royvance effort as well. Up until now, they've been primarily focused on clinical assets and taking clinical compounds through the clinic, and they actually have some approvals as well. So very strong on the clinical and commercial side, and we provide that perfect complement for the preclinical discovery side. And so we're going to continue where we are in the Boston area and Boston Seaport. Royvance is headquartered in New York, so we are going to be expanding and building out a lot of our advanced simulation efforts down in New York as well. And this just provides a huge validation for what we built, but more importantly, provides a lot of support and momentum to accelerate our vision of using physics-based simulations to overcome the key bottlenecks in drug discovery. So there's a press release there you can see on our website, and I'm happy to talk more about that if anybody's interested. Let me just play a short video just to give everybody a flavor of the types of simulations that we're running. Here we're talking about all atom molecular dynamics simulations where we account for waters, cofactors, ions, everything that's important in the system, stripping away those waters. The goal here is to understand how molecules bind to proteins, and perhaps more importantly, in many cases, how that modulates the motion of the protein. So the loop there coming in to the left, and then trying to understand how to design better molecules, not just to improve affinity, but also to improve the conformational state of the protein, especially when that comes down to things like allostery and agonism, where you have to go beyond binding and understand not just what happens to the protein, but how the consequences of the protein motion. So a lot of our advanced simulations and the technologies that we're building up are really along those lines, where binding is necessary, and we have a lot of effort and very strong capabilities in binding energy predictions, but a lot is going beyond binding energy predictions to conformational change, conformational modulation, understanding conformational mechanisms, and bringing in biophysical data, and how do we integrate all of this together so that we can get atomic resolution simulations like we were showing there, but at timescales that actually matter for drug discovery. So what are those timescales? Well, when we're talking about all atom molecular dynamics simulations, we're typically taking time steps that are on the order of a femtosecond, so 1,000 times faster than a picosecond. So femtosecond time steps, we compute all the forces between all the atoms in the system that we were seeing there before. We move all the atoms by a tiny amount, and we have to take these very small time steps so the atoms don't clash with each other, and that we can get a real meaningful simulation. And so we take these tiny femtosecond time steps, and once we have accumulated 1,000 of those, we have a picosecond. Once we accumulate 1,000,000 of those, we have a nanosecond, and 1,000,000,000 for a microsecond. So you can start to see why the heavy computing is so important, because these biologically relevant motions happen on those timescales of nanoseconds up to milliseconds typically, and that's where we're aiming at with our simulation platform. So that's looking at things like agonism and conformationally related events, allosteric binding, relative binding free energy, loop dynamics, domain breathing, and some early aspects of protein folding, although we're typically addressing protein folding and protein structure prediction from the experimental end of the spectrum, as well as some of these longer timescale events where we know that tools like NMR and hydrogen deuterium exchange can do a very good job with these longer timescale events, and bringing those together with the computation is key. We also see that water reorganization is here on the left side of this graph, and it's kind of the faster motions that we can simulate. And this was actually why we at Schrodinger started focusing very early on with WaterMap on these dynamics, with some really great work from Tom Kurtzman's group, where we started assessing the water thermodynamics associated with binding. And I'll talk about how that was such a key breakthrough in our STING project. This was probably the first real kind of industrial application of molecular dynamics in drug discovery. And the reason is because water reorganization was fast enough such that 10 years ago, we could get meaningful results before the advent of GPUs really, and the speed that those have brought. GPUs have sped up our simulations by about 3 orders of magnitude since then. So what we could typically do in a day, we could get a nanosecond of simulation time, which again allows us to look at water reorganization, and that's about it. And now we could simulate about a microsecond time scale per day per GPU, which allows us to look at lots of ligands and these motions over longer time scales. So we've built up a lot in terms of the platform. I'm not going to talk about that specifically here, just because I want to focus on the STING project and some of the higher level work that we've accomplished. But we do have a lot of details, and I'm happy to follow up with anybody anytime who wants to learn more about our efforts that we've done for building our own force field or alchemical binding free energy calculations. We've done a lot of novel work to not just compute accurate numbers, but to decompose those numbers in meaningful ways so the medicinal chemistry team and the design team can gain insights. We don't just want to have a computer running calculations that's producing numbers and telling people what to do. We want to be producing insights that allow researchers to make better decisions. And so atomic decomposition is one of those examples where with binding free energies, instead of just saying this molecule is better than another molecule by a kcal per mole, we could say it's better because of these interactions and this strain and this desolvation and again, breaking down the components. The water thermodynamics work that I mentioned with Tom Kurtzman using the grid and homogeneous solvation theory has really been provided a lot of insights. We've built up a lot in terms of virtual screening that I'll talk very briefly about, and then a lot of work on enhanced sampling and the integration of biophysical data into our simulations. So again, those will be touched on in some cases very briefly, in some cases not at all today, but I'm happy to follow up if anybody wants to get more into the details. So I'm just going to dive right into what it took to design a small molecule STING agonist. So for people who are not familiar with STING, here's a picture of 2 states of the protein, and the blue open form is the inactive state, and the purple closed form is the active state. It undergoes about a 20 angstrom conformational change. If you look at the helices at the top here, the largest motion, the binding takes place right down here at the interface. This is a homodimer, so you could think of it conceptually similar to like something like HIV protease, where you have 2 dimers coming together. The binding binding site is, or two monomers coming together to form the dimer. The binding site is at the interface of those two monomers, covered by two flaps. Again, very similar conceptually to HIV protease, but whereas HIV protease has a long, narrow binding site, that recognizes peptides, STING has a big, more globular style binding site because it binds cyclic dinucleotides. And so that means the binding site is quite large. It's also has a large degree of charge, very polar, because of binding the cyclic dinucleotides and the conformational changes that have to take place around that. So these are some of the key challenges that were associated with this target. And the approach that most researchers in pharma took were to pursue these cyclic dinucleotides. Again, this naturally binds cyclic dinucleotides, and so it's logical to say, let's just start with the endogenous ligands and make some modifications to make them more drug-like. The problem is it's very hard to make a cyclic dinucleotide more drug-like, and so most of the research went into improving the half-life from 8 minutes to 12 minutes or something like that. But ultimately, all of these cyclic dinucleotides looked like they had to go into the clinic with an intratumoral injection, and there were some efforts in the clinic to do that. Our goal was to have a systemically available STING agonist that has true drug-like, small molecule drug-like properties. So this movie is going to take us through a compilation of thousands of simulations, and just to give a sense of some of the major motions. So again, all atom molecular dynamic simulations with explicit waters, and we'll just trim away those waters so we could see what's going on under the hood here. So here's this first conformational change around the cyclic dinucleotide. And so the cyclic dinucleotide is shown there in the middle, and our goal is to induce this particular conformation of STING, which is the active conformation, but do it with a small molecule. And the challenge was overcome by thinking about doing it through a small molecule that dimerizes on itself in the context of the binding site. Now, that was initially just an idea. We modeled it in by looking at the system, and especially looking at the water thermodynamics. We identified this high energy hotspot for waters that's shown out here in red. So the red are the high energy waters. And just by building out into this region, provided our initial binding. We then noticed that there's a key PiPi stacking interaction between the molecule and itself, which was very hard to model with traditional molecular mechanics calculations. And so we actually developed a quantum mechanical method with our collaborator, Todd Martinez at Stanford, FSAPT approach that allows us to get the explicit contributions from the PiPi interactions. Then we got our first activators, and then we started looking at further improving the potency, building out into the high energy water hotspot. Started to see some very interesting conformational changes between the simulations and crystal structures. We solved about 25 crystal structures down to 1.6 angstrom resolution, so a high confidence. But we started to see some conformations like this, where this methionine loop changed conformation. We got tighter binders, but worse activity, worse cellular agonism. So we had to take a step back, redesign the molecule, drive toward these conformational hypotheses, and ultimately converge on a conformational state that resulted in strong binding as well as strong activation. And that was then what we used to drive the final aspects and the final steps of our lead optimization process. It was a highly integrated process between biology, chemistry, biophysics, and structural biology as well with the simulations, really informing each other as they go. Here's a zoom in of that high energy water hotspot, and just gives a sense of how important this type of analysis is in providing insights early on in the project. And again, this was work that we developed, the methodology and the visualization with Tom Kurtzman down in New York, and really an excellent collaborator and one of our open science fellows. Has been great to work with him. You could see the buried part of the binding site in the interior of STING is shown here. This yellow part is the initial ligand that we were modeling, and you can see there's a little pocket exiting, but where do you go if you're going to go out there? The waters tell you pretty clearly where you're going to go. You need to kind of get around this little corner and get out to this little cleft in here with the high energy waters, and that's exactly what we did. And literally in our first iteration of design, we were able to go from an idea of this dimer binding to actual binding, measurable binding affinity. We then started exploring this dimer nature, and the actual experimental data was telling us that we needed to go beyond binding and actually stabilize the dimer in a particular conformation that leads to activity. What you're seeing on the top left here, and I apologize that the font seems to be wrapped for some reason, but what you're seeing on the top left graph here is that there's actually very little correlation between the binding assays on the y-axis and the cellular signaling, the IRF signaling that we're looking for on the x-axis. And that's, again, because this is a particular challenge that goes beyond binding. We need to bind and lock the protein into a particular conformation associated with signaling. So as a frontline assay, we actually then switched from a typical binding assay to a DSF thermostabilization assay, where we're looking for the delta TM, the temperature shift upon binding. And we could see that that's a much better, there's much better correlation there between that DSF thermostabilization and the IRF signaling that we're looking for. It's of course not perfect, and you could see there's points up here, for example, on the far left, where you could have very strong thermostabilizers that have no cellular activity, and these are in fact pretty good antagonists, and these are molecules that could be taken as a completely separate project in a new direction. They stabilize the protein very strongly, but they just stabilize it in an inactive conformation. So again, the importance of really understanding not just the binding event, but what happens beyond binding. And another very important part of this project was that we wanted to bind to the wild type form of the human STING protein, as well as the highly prevalent HAQ allele, and also the relevant efficacy in tox models. And we could see here that our molecule SNX281, shown in cyan, has roughly the comparable thermostabilization to the 2', 3' CGAP, the endogenous cyclic dinucleotide, across all forms of the protein that we care about. Very interestingly, people that are familiar with STING might know of this molecule DMXAA. It was a very encouraging molecule found a few decades ago through a phenotypic screen and they found that it worked really well in mice, and the preclinical data was actually quite compelling. And this molecule was taken into the clinic. You could see it stabilizes the mouse form of the protein a bit, but it does nothing to the human form. And this molecule didn't make it through the clinic because of lacking efficacy. So just, a little lesson up front that, pay attention to not just the animal models, but obviously really try to understand how that's going to translate into and through the clinic. I also mentioned this PiPi stacking interaction that we developed. This was in collaboration with Todd Martinez, and it's actually really one of the points to highlight, and that's the importance of having a team of experts that can develop methods as needed. In experiments, we always think about this, that we need a team that can develop custom assays, but often computationally, people are using packages of kind of precompiled software, and you deal with what you get. And in these more challenging drug discovery projects, there's often these edge cases and corner cases that don't fall into the bucket of the standard software, and it's important to develop methods as you go. So I mentioned this PiPi stacking interaction, which was really critical for this thermal stabilization and getting this molecule to come together and dimerize in the context of the protein. PiPi stacking interactions are not modeled very well through traditional molecular mechanics methods, and very hard to intuit, even if you're just modifying a ligand that's interacting with a phenylalanine to the protein, for example. But in this scenario, the molecule is a dimer. You modify one side of it, the other side's changing. So there's just a lot of moving parts. It's very hard to gain any intuition around what would stabilize that interaction. So we developed this functional group-based symmetry-adapted perturbation theory with Todd Martinez, who is the author of the TerraChem quantum mechanics package. And in the course of about 2 weeks, we were able to implement the method and validate it and deploy it on the project and overcome this bottleneck. And you could see quite a good correlation for these earlier modifications. This is a very relatively small number of modifications they were making on the ring, very small modifications, mostly slight electron pushing and pulling groups. But we could see we were able to go from this early hit to okay and ultimately quite potent molecules just by tuning the nature of this PiPi stacking interaction. And then a lot of the project was computing binding free energies. We've developed quite an extensive infrastructure for relative binding free energy calculations and absolute binding free energy calculations using alchemical free energy methods. And so this is similar to an approach like thermodynamic integration or free energy perturbation, FEP plus, people might have heard of. So that's the approach, loosely speaking, that we're taking here. High quality force field, all atom molecular dynamic simulations, and a rigorous calculation of the difference between different molecules in terms of binding. And what we could see here in this graph on the right is the distribution of the molecules that we predict to be good, which is the blue curve, is much better than the distribution of the molecules that we predict to be bad, which is red. And so you could just think of this in terms of, you come up to make a decision in terms of chemistry. You want to figure out what's the next 5 molecules to make. You run 100 molecules and you look at the distribution of what are the molecules we say that are good, what are the molecules we say that are bad, and how do those actually turn out experimentally, which is shown here on the x-axis. And so what I like about graphs like this is it shows the real distribution. It doesn't just say that the error is 1 kcal per mole or not on average, because that doesn't give you as much information about the whole picture. What we see here is the calculations obviously are not perfect. These are still models. If we had to choose from one of these distributions, we would, and we want to make our molecules better, meaning, more favorable, more negative, we'd obviously all choose from the blue distribution. But there's molecules that we say are bad that are actually good. There's molecules that we say are good that are actually bad. And a lot of these molecules we want to make anyway because we learn from it. And so we're not, again, just using the computers to tell us what to do. We're using the computers to gain insights and continually improving the methodology and the protocols as we go through the discovery process and get data back. In total, we only had to make about 200 compounds here. And so even though we ran about 20,000 relative binding free energy calculations over the course of this project, we only made about 200 molecules to get to our development candidate. And I touched on a little bit about the use of biophysical data in our simulations. For this particular project, we used some NMR and small angle X-ray scattering data. We've more recently in some of our projects started working in hydrogen deuterium exchange and are exploring other experimental data sources to augment the simulations. And this is some work from our CTO, Huafeng Zhu, that he published in Journal of Chemical Physics. And you could read about the details, but the big picture is that we could take our original potential, which is basically what our molecular mechanics force field would tell us to do. And then we could augment that with biases of various flavors based on the experimental data. And this is something that I think is really a huge contribution to the field and something that we think is going to help accelerate drug discovery on these harder cases where simulation alone or experimental biophysical work alone are insufficient to give a high resolution view as to how the protein actually works. And maybe more importantly, the most biologically relevant motions of that protein, which would then allow us to focus our simulations on the motions that matter. And that's all active development work that we're continuing to integrate other biophysical methods. As far as our overall design process, we start with ideation. So this is not just a bunch of computers that are, spinning and coming up with ideas. It's still people that are coming up with ideas, talented, experienced in-house medicinal chemists who are looking at the water thermodynamics, who are looking at the atomic decomposition, who are looking at the conformational modulation of the protein. And all of these ideas then come into our design meetings where the medicinal chemists and the modelers and simulation leads are all sitting down trying to figure things out. And then this is also where we pull in the software engineering and research programming team as necessary, if there's particular bottlenecks to overcome. So for example, when we realized that we needed to account for that PiPi stacking interaction, we had to bring in the quantum mechanics development team to come up with a solution for that. And we're doing this routinely. The method developers have long-term projects, obviously, but day to day, whatever the project encounters is the most important problem that we're trying to solve. So out of this meeting comes our recommendations for molecules, 100 or hundreds of molecules in many cases that we run through the simulations. And that's typically computational binding free energy assays, as well as these conformational free energy assays, which leads to ultimately the selection of the molecules that we're going to synthesize, generate experimental data, and iterate on that. So nothing really substantially different than the typical drug discovery cycle, other than that we're very heavily informing our ideas and our selection based on physics-based simulations. So a few snapshots of the experimental data. What we're looking at here is the signaling of our compound SNX281, shown here in blue, as compared to the endogenous cyclic dinucleotides, 2', 3' CGAMP in this case. And we see that in all cases with the wild type, the HAQ allele, and another allele, we're seeing that we get more potent stronger binding and more potent activation across all three of these cases here. And what this ultimately leads us to, and sorry for skipping over a lot of the other experimental data, but ultimately what this leads to is complete tumor regression after a single dose. And so what we're looking at here in a CT26 mouse model, we're looking at a single dose shown here on the left, and the curve, which is 8 members of the cohort in cyan, basically all 8 of them completely cured with a single dose, and the rest of them, the tumor continues to grow. Perhaps even more interesting is the fact that there's a tumor memory. And so when we take those cured mice that were cured after 30 days, we continue that, let them go for 90 days. And then what we do is we reintroduce the tumor into those cured mice, as well as reintroduce it into 8 previously untreated mice, and the previously 8 untreated, all the tumor grows, of course, but in those 8 previously treated mice that all recovered and were perfectly healthy, with no additional administration of SNX281, the tumor doesn't grow back. And so there is indeed a memory of how to deal with this. And that was the ultimate goal of this project, which I probably didn't say clearly enough at the beginning, but using STING to spark the innate immune response and cause tumor infiltration by lymphocytes to get rid of it, and that's what appears to have happened on the left. And again, that memory, that transfer from the innate immune response to the adaptive immune response appears to be quite robust. Still a lot of very interesting biology and work that we're expecting to find from our clinical trials, but extremely exciting area of research if you think about the possible consequences of an innate immune modulator like this for cancer. So that was a summary of the STING project, and now I want to get into a bit about our conformational genetics work, how we think about targets and select targets and use the physics-based simulation platform to understand conformational modulation. So the basic workflow, we start with human disease-driving genetics. We map those genetics onto structure and try to assess druggability and the conformational behavior of those proteins to come up with an actual design strategy. As one example of this, SHIP2 was a project that we started early on and decided to really focus on it for validation as opposed to drug discovery. There were already fairly advanced programs from Novartis and Relay and several others, but we thought it was a great target to work on developing this approach. So if you look at in the sequence space and you look at these kind of hot spots for glioma, you see that there's a couple regions here circled on the top left that are quite disparate in sequence, but when you map those onto the structure in 3D, you could see they're all right at the interface, and these are all right at the interface between the phosphatase domain, kind of on the bottom left, and one of the SH2 domains shown on the top right. And so the idea here is if we could understand how these mutations are working, we could come up with a way to drug the target. And so what we wanted to do is look for the conformational change associated with these mutations. The time scale to see these large-scale conformational changes was beyond what we could do with a standard molecular dynamics approach. So we had to move toward a enhanced sampling approach. And so what we did in this particular case is we kind of in a cartoon representation tied a spring to the SH2 domain and started pulling on it to see the consequences of pulling that. And we could do that in the context of the mutations. We could also do that in the context of a glue molecule that's represented here in yellow. And then that would allow us to then measure what we believe matters in this case, which is the protein going from an inactive state, an autoinhibited state where this SH2 domain is blocking the catalytic activity of the phosphatase domain, to an activated state where that SH2 domain has been dislodged. Again, these are all gain of function mutations that we're seeing right at that interface. And so what we could do is we could simulate all of those mutations. And this is the type of simulation that we're talking about here for one of the scenarios. And looks like it might have froze for a second. But if it catches back up, it'll start moving again. And what we see is that the protein can start to dislodge, the SH2 domain dislodging from the phosphatase domain, and we can measure the amount of work that it takes to dislodge that. And we can do that in the context of different mutants. In this particular case, we're just showing one of the gain of function mutants, but we've done that with all of them. And we could see relative to the wild type protein, which is shown here in gold on the top right plot, the gain of function mutant is actually destabilized. It's easier to pull it off, which makes sense. That's why it's a gain of function mutation. It destabilizes this autoinhibitory state. And then we can start to look at what happens if we put different ligands in there, different putative drugs in this region here to act as glue to restore or stabilize this inactive conformation. And we're able to make those predictions, and we were able to drive this project over a log in potency in just a few iterations using this approach. And this is actually how the Novartis and Relay molecules work. They're these allosteric stabilizers, which, as we're probably mostly familiar with, targeting the catalytic site of phosphatases has been a huge challenge and continues to be a challenge. But this allosteric approach of locking the protein into an inactive conformation is quite compelling, and is likely that it could be applied to other phosphatases as well. So that's one particular application of this conformational genetics, again, understanding the conformational behavior and especially using human disease-driving genetics to understand that behavior, developing a computational protocol to predict the consequences of those mutations, then using that to come up with a strategy to drug the target. Here's a more exploratory target, NLRP3. It's part of the inflammasome. So what happens here is the NLRP3 is a fairly large protein on its own as a monomer, shown here on the left. But this purple domain undergoes a conformational change upon activation. So it goes from this inactive state, where this purple domain here is on the right, that moves out of the way to the left. And what that does is it allows for dimerization across this interface, and ultimately this oligomerization, forming this 11-mer, which is the inflammasome. And that's then what kicks off the inflammation response. So one could think about ways of inhibiting the inflammasome, but dealing with something of this scale with a small molecule is very hard to wrap your head around how one might do that. On the other hand, one could imagine maybe we could just lock this protein into this inactive conformation. So the question is, can we understand that inactive conformation and the conformational mechanism associated with that inactive to active transition? And if so, can we come up with a plan to drug it? So these were more advanced simulations. This is later. So rather than just pulling and seeing that one transition out, now we've developed a new methodology called meta-EABF. It combines metadynamics and is an extended adaptive biasing force approach. And we could sample multiple configurations going from active to inactive. And what we see here is that that the wild type that's shown in purple on the graph there's still a barrier that going from that inactive starting state to that active state. But in the context of all the gain of function mutations, they all destabilize that inactive conformation. So that gives us a lot of confidence that the disease is in fact driven by this destabilization of the inactive conformation, and then guides us toward how we might drug it, which is to stabilize this inactive conformation. So even in the context of mutations or diseases, that we could restore the inactivity and reduce inflammation. So we surveyed the protein. We looked for binding sites around this interface. In fact, we found what appears to be quite a nice binding site at the interface between two of the domains. We did some druggability assessment, and we've developed a bunch of druggability tools. We found that there appear to be quite druggable pockets right there at the interface. We did a large-scale virtual screen, and I could just say in a few words, our virtual screening approach, which is starts off with a pretty standard large library, tens of millions of compounds commercially available, and we prepare those, filter out molecules based on whatever our preferences are, and then go through a large scale docking experiment where we're docking tens of millions of molecules using a combination of different approaches. And the goal here is not to get the right answer, but really just as a filtering step, slightly more sophisticated filtering than just the property filtering. But still, for the most part, we're not counting on docking to give us the answer, especially in these harder systems with induced pockets and sites that haven't been drugged before. So docking helps us filter things down, and we try to, at the end, get out a few tens of thousands of molecules that we take through molecular dynamics and absolute binding free energy calculations. And this is then what's really leading us to our decisions of what molecules are better or worse, because now we're accounting for all the waters and the protein dynamics and the entropy and desolvation and what we feel is needed to have a rigorous assessment and a high confidence prediction in the ability of these molecules to bind. At the end, we still sit around with a bunch of chemists and modelers and designers and look at the top 1,000 or so molecules to make our final selection of a few hundred molecules. But this process as a whole has proven to be very useful in our efforts because we get such richer information than you would get from docking alone. So what came out of this was, a bunch of hypotheses. We actually have not decided to pursue this target yet. We've been focused on oncology up until now, so we have not gone into inflammation. So this was really an exploratory project, but we came up with a bunch of molecules that came out of the screen that appear to bind well based on the binding free energy assessments. And when we do the atomic decomposition, and we could look here at these, the green mesh spheres, we could see that we're getting interactions from two different domains, which is exactly what you would want to produce this glue style mechanism to hold things together in the inactive conformation. And then we've reperformed those meta EABF calculations, and with these molecules in place, pulling on those two domains increases the barrier to activation considerably. So in conclusion, I think, hopefully I've explained that we've built a fully integrated chip to clinic company at Silicon Therapeutics, an industry leading platform for running these molecular simulations, and especially the more advanced, complicated simulations. Having a team to develop custom apps is key for drugging challenging targets. Integration of biophysical data has helped us generate more accurate and more biologically relevant simulations. And the small molecule STING agonist program, I think it really shows what can be brought to the table with this physics driven approach. So with that, I'd just like to wrap things up. We have a lot more information on our website. We've posted a nice little video on YouTube that describes things briefly. We've built an awesome team. We're right down in Seaport. Would love to have anybody visit when we get back to normal non-COVID times and check out our operations. We are hiring heavily right now across the board, chemistry, biology, biophysics, as well as advanced simulations and the rest of the platform team. So feel free to reach out to me or just go to our website if you want to learn more. Most of the positions are not posted yet. There's going to be a lot of them coming soon, but feel free to reach out to me and I'd be happy to tell you more about what we're doing. And with that, happy to pause and take questions.
Great. Thank you very much. That was a fantastic talk. Questions? We can unmute so people can discuss. There's a reaction button. You can raise your hand and I can call on you. You can also send questions to me by chat. So let's see, allow to unmute. I think people should be able to unmute themselves now. I see a couple of hands coming off. I've got one to kick it off. Just from the STING story at the beginning, you're targeting that conformational substate in this, and you showed that you can target multiple states that, are not created equal in terms of activity, right? So is that conformational state something that you see experimentally, like get lucky in a crystal and you've got that one, or maybe by NMR?
Yeah, well, so in some sense, we kind of knew the answer up front because we knew that the endogenous cyclic dinucleotide activates STING, and so and there were crystal structures already of these cyclic dinucleotides bound. So that definitely gave us some guidance. We had the apo structure in that inactive conformation. We had the cyclic dinucleotide bound structure in that active conformation, but most of the small molecules were not able to induce that CDN-like active conformation. So the goal, so we had a very good sense of where we wanted to get to, because that's ultimately where we wanted to drive things toward that cyclic dinucleotide state. In the end, we ended up finding molecules that stabilize that even further and created a stronger activation than the endogenous ligands, which is what we were looking for. So there were some subtle refinements at the end, really iterating with the biology and the structural biology to converge on some slight variations. But overall, we had a good idea at the beginning. We wanted to induce the CDN bound state, which was somewhat well known, and the challenge was just how do we get there? Binding alone wasn't telling us that, so it was binding plus the simulations that was guiding us toward that conformational mechanism.
All right, great. Bharat, go right ahead.
Hi, great talk. You mentioned you use quantum mechanics in your actual simulations. Do you actually do that for the whole protein, or do you just focus that on just the binding site?
Yeah, definitely the latter, as well as in our force field parameterization. So every molecule that we do any kind of simulations with, we fully parameterize it using quantum mechanics beforehand. So we've developed a engine where you send in a molecule or hundreds or thousands of molecules, and we do a full quantum mechanical torsional scan around each rotatable bond, as well as an electrostatic potential fit. So we're constantly running quantum mechanics to get the best force field parameters for our small molecules. And then we're also using it in these one-off cases, like I showed with this FSAPT approach for PiPi stacking, or if there's other nuanced interactions, and then we look very specifically at those interactions and augment those. Quantum mechanics is just still much too slow to run on protein-sized systems, and definitely much too slow to run with protein-sized systems with water and everything. And the primary gap most of the time in terms of accurate predictions in these biomolecular simulations is the sampling itself. And so if you had to choose between more accurate scoring models or more sampling, typically more sampling is where you want to put your effort, and that's where molecular dynamics comes in. So molecular dynamics run long enough with an accurate force field is good most of the time. And then to tease out some of these more subtle interactions, pi-pi, pi-cation type interactions, and we augment with the quantum mechanics.
Thank you.
Suvaroy, go right ahead.
Hi, am I audible?
Yes.
Yep. Sounds good.
I was curious to know, like, if you replace the dinucleotide with a dipeptide having two aromatic residues, do you have the same kind of effect on staying, or is it different?
We have not done that. My guess is it wouldn't bind. So you're talking about it would probably have to be a cyclic peptide, I would guess. I don't think you could form a 2 residue or 3 or even 4 residue cyclic peptide. It would have to really fit right in there. So getting the charges right, I don't think would work. So we didn't attempt to do anything like that, but my sense is a peptide wouldn't do anything.
Okay, and is it possible that is there any oligonucleotide or peptide that would completely disrupt the dimer interface?
Quite possibly, and that's what one would look for as an antagonist, and that's something that we didn't pursue. We were focusing completely on cancer and oncology, but for inflammation, I mean, STING, for example, is a potential lupus target. A lot of other inflammatory diseases where we think a STING antagonist would be important. And one approach would be to disrupt the dimer interface. And so whether it's disrupt the dimer interface or just bind, some of those molecules that I showed that bind but don't stabilize, or stabilize but don't activate, those types of molecules would also be potentially interesting. I think that's a really hot area, and I'm hoping that there will be some medicine starting to enter the clinic, because it is such a central target in the innate immune response, and we feel strongly that a good antagonist would have a very strong opportunity in the clinic.
Okay, thank you.
All right, Pete, go ahead.
Thanks. Thanks, Woody. That was great talk, covered a whole lot of area. I have three questions on three completely different subjects. So I guess the first one is kind of related to what Bharat was asking about. Do you have any perspective on the multiscale molecular dynamics simulations to try to use like a coarse-grained model for some areas and possibly a finer-grained model for areas of interest? And if the complexity of that is worth looking into versus just burying it with compute power?
Yeah, so we are definitely looking into multiscale modeling and coarse graining and stuff like that. I think there is a role for those types of simulations, especially when trying to understand biological mechanisms and some of these much larger scale conformational changes. For example, we're looking at targeted protein degradation, and to understand the ternary complex with the E3 ligase is one thing which we could model atomistically, but when you start talking about the whole Cullen ring system and everything else and how that whole ubiquitination process happens, that's just too large for us to be doing routinely with all atom molecular dynamics simulations. So I think there are roles for coarse graining. There are roles for mixed style simulations as well, if you want to start getting into like QMMM type of approaches. But you start to lose a lot in terms of speed and sampling, and dealing with the interface region starts to become pretty fuzzy. There hasn't been any good evidence, to my knowledge, that those types of models get you to be more accurate. And typically our bottleneck is getting accuracy at a resolution where we can make confident predictions. And so we need to get to accuracies of essentially a kcal per mole, because the molecules that we're designing to say molecule A is going to be better than B is rarely going to be much different than a kcal per mole or two. So if we can't get at that resolution, then the method is generally not going to be that helpful for decision making. So these coarse grain models, I think, will be informative for understanding biological mechanisms, and they probably will help us structure our experimental design, but probably not so much in terms of the quantification of a particular molecule being better or worse, and how to design it. The cost, just when you start doing quantum mechanics, the costs go up so fast. I mean, it's not like it's a, to do the simulations that we're running now, where we're running a microsecond of simulation per day, for example, on a GPU, to run that same size system with quantum mechanics would take the whole computing power on Earth, and it would take probably thousands of years with that to run the same style of simulation. So QM alone can't really be approached. These kind of mixed resolution methods with QM are interesting. We've been taking a pretty simple approach, which is just doing the all atom molecular dynamics simulations and augmenting where we think it's important, just because there's been a lot of effort going into these QMMM methods and hybrid methods, and no real evidence to date that suggests that they're better. One day they will be, but I think right now they're still just too limited by sampling. And if you're not sampling sufficiently, then it doesn't really matter how good your energy model is. Yeah.
Thank you. I hadn't heard anything new about them either, but your perspective is much more up to date than mine. And thank you for explaining that.
Absolutely.
Second question, when you were talking about your CT26 PD1 story, for using autoimmune response as a cancer therapy, or for using innate immune response for cancer therapeutics, is that too early stage to be worried about triggering autoimmune responses similar to like AIDS or not AIDS, MS or lupus? I apologize, I can't talk today and I'm mixing up all my phrases.
No problem at all. There's no indication that this molecule clears extremely quickly. It doesn't, not as fast as the CDNs, which clear in the course of minutes. Our molecule clears over the course of hours, and so it doesn't hang around at all. I don't see any implications, and I don't think there's any biology or genetics that would suggest anything like that. The main concerns are that there's the cytokine response, and you've got to balance out the the interferon response that you're looking for to activate the innate immune system, and the pro-inflammatory cytokine response, which you want a little bit of that because you want to infiltrate the tumor cells, but you don't want too much of that. And so that's basically the primary focus of the dose escalation that we're in right now, is understanding exactly when that kicks in. There's antidotes for cytokine storms, so we're prepared for that, and we just have to titrate and figure out exactly where's that sweet spot where we get the interferon response that we're looking for, without the cytokine response that would be too inflammatory. So that's really the balance. I don't think there's any real concern with autoimmune diseases, although if you are familiar with it or anybody is, please let us know, but I don't think there's any evidence for anything like that.
I don't either, but I know some people that do have autoimmune disorders, and so that's always a question on my mind with these types of therapeutics.
Yeah, absolutely.
Yes. Last question I've got is and this is a very low-level question, but for the different types of simulation that you're doing, the kind of steered ones versus the free ones, is there a difference in the number of replicates that you have to do for those types of simulations for you to be comfortable with the results?
Yes, absolutely. Yeah, great question. So for our typical binding free energy calculations, we run everything in triplicate. And so, we get, error bars, they tend to be pretty tight. And, when one of them's off, that typically is an indication that the system hasn't converged or something's been set up incorrectly. So it helps us dive deeper into the problems. For these longer timescale simulations, like you were seeing with like NLRP3, that we typically do 30 to 50 replicates to get, just much more sampling, much better statistics. Obviously, a much more complex free energy surface that we're trying to converge. So the accumulated time that we run for binding free energy calculations is, on the order of a few hundred nanoseconds. To converge those is typically on the order of a few hundred microseconds with enhanced sampling. And so, yes, a lot more replicates gives us a lot cleaner curves. If we're only running 5 or 10, we can make crude predictions, but we've found that typically it's on the order of 30 to 50 to get confidence on the order of a kcal or 2.
Thank you.
Yeah, excellent questions. Thanks.
All right, great. Well, if there's no more, I have one more, and then we can kind of wrap up. And maybe it's I don't know much about the field, but this dimeric ligand, how common is that? If that's and is this guy is it dimeric in the binding site, or is it dimeric in solution also, and then it just binds as a dimer?
Yep. So it's quite unique. When we started this project, there was nothing like this in the PDB, which obviously makes sense as to why there were no programs and tools out there to do things like this. And so, we had to develop that FSAP program. We also had to develop our own docking routine to do this because, you have 2 molecules coming in together. Every docking program just does 1 molecule at a time, and you can't do that like that. So we had to develop a lot of tools because there was nothing out there, because it's never been seen before. There's one case, if you go through the PDB, one case that I found where you look at a ligand interacting with itself, and that was just through a crystallographic interface, and that does happen to some extent, but nothing with it really biologically relevant stacking on itself. Since then, and unbeknownst to us at the beginning of the project, Merck actually was pursuing a similar approach to this, and they ended up pushing their cyclic dinucleotide into the clinic and not their small molecule. I think it just didn't have quite as good of properties as what we developed, but they did publish that, a very nice paper. I believe it was Nature Methods last year, if not Nature Methods, might have been one of the other Nature journals, but very much worth looking into. It's a beautifully written paper, and they characterize this whole dimer nature very explicitly. They use NMR, and they find that it pre-dimerizes. And so you have, the molecule is highly soluble, but there's a small fraction that pre-dimerizes in solution through that PiPi stacking interaction, and that's the entity that binds. One could imagine it binding one and another successively, or kind of pre-forming and binding. Either way, the binding site's very flexible. It would need to open up considerably to even let those things in there. So we have not gone down those deeper explorations because we're worried, because it's very interesting, but it'll take a lot of effort, and we need to keep focusing on our drug discovery. But at the same time, it is a super interesting problem, and we would love to get deeper into it. And if there are people that would like to collaborate on something like that, it's quite I think, novel and unique mechanism, but the fact that Merck and Silicon converged on that independently suggests that it's real, and there's probably other targets where you could start to imagine getting away from these kind of very large endogenous ligands and using this type of approach.
Yeah, that's it's super interesting. I'm thinking about it when you showed the simulations, sort of like a mechanistic on rate, and then when you try to think about, okay, well, this thing has to pre-dimerize into the right conformation because it's probably pretty greasy and it sort of slides around, and then that contributes. That it's an interesting problem.
Yeah, totally. Yeah, fascinating. And we were pretty shocked, happily so, that this initial hypothesis did end up panning out.
That's great.
Yep.
All right. Well, with that, Woody, thank you very much. Fantastic to hear about all the exciting work that's going on there. And thank you, everyone, for tuning in today. And be sure to catch us next week for our next SBGrid weekly webinar.
Yep. Great. Thanks a lot, Jason. Thanks, everybody.
Take care.
Take care. Yep. Bye-bye.